Hey, Hammy here with part 2 of section 7.2, on which we want to look at the genetic code and also the process of translation, how that protein is actually made off of that mRNA transcript. One of the first things before we get into the process of translation that you need to understand is how the actual genetic code is read. Okay, the genetic code is in a triplet code, which is 3. So 3 nitrogenous base pairs at a time. Each group of 3 on the DNA, which is coded then onto the mRNA, is called a codon. Okay, so it's the code. The code is a triplet. Each codon then specifies an amino acid. Okay, so if you have 4 bases, A, T, G, and C, okay, and you are using a triplet code, so 3 at a time, you get 4 cubed, which gives you 64 possible codons. So we often use kind of a decoder type chart over here, where you start in the center. So let's say you have the codon DNA's TAC. Well, first you have to transcribe that into mRNA. So the T goes to an A goes to T, but remember on mRNA it has uracil instead of thymine. So AU, G, C, T goes to A, G, 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 U, U, A. Okay, so this is going to be our mRNA transcript, okay, transcription. When that's taken out to the ribosome and read, okay, it's going to read these codons 3 at a time, and it's going to bring in the proper AA or amino acid to build the protein, because remember, amino acids make up proteins. In order, if you have a round circular chart like this, and I'll show you a different type here in a moment, we start for the first one is AUG, the first letter A. You find A in the center, in the very center circle here. So you have A, and then you go to U, and then you come down here to G. Okay, so our first amino acid is going to be methionine. We often abbreviate them with the first 3 letters. Okay, then CAG, we find C, come out here to A, and then find G, okay, which is the amino acid GLN. Okay, next GGU, GGU, which is glycine, GLY, and then UAA, UAA. Huh, what's that one? What are these little circle things here? Okay, that is a stop. Okay, so you've got start and stop codons. Notice the triangle down here by methionine. Most genes or proteins will start with the amino acid methionine, and that's often called the start codon. And it will read along the mRNA until it gets to AA or UAA, UAG, or UGA. And these circles over here, okay, here, and here are the stop codons. And then that tells the ribosome to stop making the protein, to stop reading the mRNA. Okay, there's 3 characteristics of the genetic code. Number 1, it's universal. In other words, all living things, bacteria, fungi, plants, animals, okay, we all use these same codes. Okay, so when CUC, okay, is all, we use the same 4 base pairs, A, T, G, and C in DNA, and then A, U, C, and G in RNA. And there we have very similar amino acids and proteins. That's why we can eat food, and we take those proteins in our food and break them down into amino acids, and we can use them to build our own proteins in our own cells. The genetic code is unambiguous. Each codon, CUC, is always going to be leucine, okay? Always. It never changes, okay? And it's going to be, number 3, it's going to be redundant. You'll notice that's not the only leucine, okay? There's only, when we talk about amino acids that we need, there's only 20 on this chart, okay, plus the stop codons, okay, and we can build all of our proteins out of those 20 amino acids, okay, in varying lengths and so forth, like we do with we have 26 letters in the English alphabet, and think how many words we can make because you put them in different combinations and different lengths. Same thing with 20 amino acids that can make thousands of proteins. Okay, but notice it's redundant. In other words, CUU, CUC, CUA, CUG, UAA, UGG all make leucine. Okay, so there's more than one amino acid that can code for an amino acid. Okay, now CUC, remember, it's unambiguous. It's always going to be leucine, but leucine can have multiple codons that code for it. Okay, now onto the process of translation, where we're going to take that code on the mRNA and turn it into a protein. Okay, again, this is happening in the cytoplasm where you find the ribosome. Okay, so the mRNA leaves the nucleus after it has been edited and spliced and all that we talked about in the previous video. Okay, and it's going to be made into the protein. In order to do that, you're going to use a ribosome, which I'll show you here in a second, and the third type of RNA, tRNA, which stands for transfer RNA. Okay, transfer RNA kind of has this cloverleaf structure when it's kind of drawn out like schematically. When you do the shape filling model over here, I almost think it looks like a triangle or pyramid or boomerang or something. And at the bottom, down here, you're going to have what's called the anticodonin to the codon we just talked about on the mRNA. So there's going to be these groups of 3. Each one is going to be a codon, 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 codon. Okay, and these things are going to, the anticodons are going to match up with the codons, and up here it's going to attach the amino acid right here. The attachment site is at the top. Okay, so over here you can actually kind of see it. Here's where it's going to attach. And you'll see in the next slide that this thing it's called transfer because it goes out in the cytoplasm, picks up the right amino acid, and then brings it to the ribosome, and it plugs them in the correct order. Okay, it reads the recipe and puts the stuff together in the right order. Okay, the actual steps of translation. Okay, first the mRNA attaches to the ribosome. So here, this kind of brownish, tannish blob here is our ribosome. Okay, the mRNA is this sort of light green strand that's laying in there. Okay, it's going to attach to the ribosome, and then we'll show some animations of this in class. But what's going to happen? It's going to read 3 at a time. So the first one, UGG, okay, was where this tRNA was here. So look, anticodon. U goes to A, C to G, C to G. And now notice we use U's because it's tRNA, so it's still RNA. It's going to have uracil instead of thymine, okay? So ACC or UGG is the codon for tryptophan. So you can see this one had brought in tryptophan. The whole thing slides over, okay? So the tRNA picks up the amino acid. The anticodon matches up with the right codon. It moves into position. The whole thing slides over. The mRNA moves along the ribosome. Okay, the next codon, UUU, okay, anticodon UUU matches up with the codon AAA, brings in lysine, okay, brings in lysine, okay, and then the next one, GAU, okay, codes for aspartamine, okay, which the anticodon is CUA. So these things just keep moving over, and every time they do, you see this little line right here? Okay, I want you to make a note of that in your notes. It's called a peptide bond. Okay, a peptide bond forms between these amino acids, and they just keep connecting together, connecting. And as the ribosome and the mRNA move along, okay, as the mRNA slides through the ribosome and it's read 3 at a time, 3 at a time, 3 at a time, it keeps bringing in amino acids, and as the whole thing shifts over, you get this growing chain of polypeptides. Okay, or what we would call a polypeptide, which is the beginning sort of primary structure of a protein. And it just keeps going until it gets to a stop codon. Okay, once it gets to the stop codon, okay, this ribosome will fall off. The ribosome will fall off, and you have sort of your leftover polypeptide chain. Here's kind of a good pic that gives you sort of an overview of the whole central dogma. Okay, we're starting with the genetic code. Here in the nucleus is the DNA. It doesn't leave, it's protected here. Okay, then it is transcribed into RNA, which is then edited. Okay, leaves the nucleus. Right here's the nuclear membrane, which has pores or openings. It's taken out to the cytoplasm. Okay, so our mRNA transcript is taken out to the ribosome, where translation takes place. Okay, and here you see the tRNAs bringing in, okay, over here, bringing in the amino acids, okay, these little balls on the end here. And as this thing slides along, okay, this chain right here is going to keep getting longer and longer and longer. Okay, when it's done, okay, then remember it goes into the rough endoplasmic reticulum, which is then taken to the Golgi apparatus, okay, where it's that final shape of the protein is made, that three-dimensional shape, that secondary, tertiary structure. Sometimes the quaternary structure, like hemoglobin, it might attach, you know, two other polypeptides to it. And it's packaged up, and it will either be delivered somewhere else in the cell, or it can be packaged up for excretion. Maybe it's a digestive enzyme, and it needs to be secreted into your small intestine. So just to kind of give you an overview of the whole central dogma of how we go from the code on DNA to the actual protein out here.